Hey everyone, welcome back for another deep dive. You ready to explore some more fascinating microbiology?
Absolutely. I'm always up for a journey into the microbial world.
Today we're going deep into the fascinating world of antibiotic biosynthesis. It's a topic that's not only super interesting, but also incredibly relevant for medical microbiologists.
Oh, absolutely. Especially in this era of growing antimicrobial resistance. Understanding how nature makes these powerful compounds is more crucial than ever.
It's like we can peek into nature's laboratory and learn some tricks of the trade.
Exactly. And to guide us on this journey, we're diving into the book Antibiotics: Challenges, Mechanisms, Opportunities by Walsh and Winsvich.
Specifically, Chapter 14, Antibiotic Biosynthesis. Principles. It lays out the foundation for this amazing field.
It's a treasure trove of information.
All right, so let's start with the basics. Just how many natural antibiotics are out there? It's got to be a huge number, right?
You're not kidding. By 2012, researchers had already identified an astounding number, something like 70,000 different microbial natural products.
70,000. That's
Yeah.
mind-boggling. But are they all antibiotics?
Not all of them, but a good chunk. About half exhibit some form of biological activity, and around 16,000 have known antibiotic properties.
Wow. So we have this massive library of potential antibiotics just waiting to be explored.
It really highlights the incredible potential of microbes as a source for new drugs.
And to think a big portion of the antibiotics we use today come from just one group of bacteria, the actinobacteria, particularly those from the genus Streptomyces.
Streptomyces are like the rock stars of antibiotic production.
But here's the kicker. Scientists believe there's a vast, untapped reservoir of potential antibiotics hidden within these microbes, a potential 25-fold increase from Streptomyces alone.
A 25-fold increase. Now that's what I call a hidden treasure. But where are these antibiotics hiding? Are they like secret agents waiting for their mission?
Well, they're not exactly hiding. They're in the genes, but they're usually turned off. These cryptic gene clusters have the blueprints for making antibiotics, but they're not active under normal lab conditions.
It's like they have this incredible potential, but they're just not showing their cards.
So how do we tap into this potential? How do we awaken these sleeping giants?
Researchers are getting pretty creative. One approach is co-culturing, putting different microbes together and seeing if they can trigger each other to activate these silent genes. It's like setting up a microbial party and seeing what happens.
Microbial matchmaking. I like it. And what about synthetic biology? Is that being used to manipulate these gene clusters directly?
Absolutely. By understanding the genetic code, we can try to flip the switch on these genes and see what amazing compounds they produce. It's cutting-edge stuff, and it holds incredible promise for the future of antibiotic discovery.
It sounds like we're on the verge of a whole new era of antibiotics. But before we get too carried away, I think it's important to understand how these microbes regulate their antibiotic production in the first place. It seems counterintuitive to even have these silent genes. Why bother?
Well, antibiotic production is a complex process, and microbes have evolved sophisticated ways to control it. It's not a make it all the time kind of situation. It's all about efficiency and responding to the environment.
So it's about strategy. It's about knowing when to deploy those antibiotic weapons.
Precisely. And to really understand this, let's look at our superstar Streptomyces. It's famous for producing streptomycin, but did you know it has the potential to produce at least 22 other antibiotics?
22. It's like a microbial pharmacy. So how does it decide when to activate one blueprint over another?
It all comes down to a fascinating cascade of molecular events. Think of it like a chain reaction, where one step triggers the next.
Okay, lead me through this chain reaction. I'm ready.
Well, it starts with a molecule called A factor, which is a type of butyrolactone.
Butyrolactone. Now that sounds a bit technical. What role does it play in this whole process?
Think of butyrolactones as Streptomyces hormones. They act as signaling molecules influencing things like development and, importantly, antibiotic production.
So A factor is like the messenger kicking off the whole antibiotic production process.
Exactly. And this messenger interacts with a repressor protein called ARPA.
Repressor protein. So it's stopping something from happening.
Right. When ARPA is bound to the DNA, it prevents the production of another protein called ADPA, which is an activator.
Okay, I see. So we have A factor, the messenger, ARPA, the repressor, and ADPA, the activator. What happens next?
When A factor binds to ARPA, it releases it from the DNA, allowing ADPA to be produced.
So A factor is like the key that unlocks the repressor.
Exactly. And ADPA, the activator, is pretty busy. It activates the transcription of yet another regulator called STI-R.
Okay, we've got A factor, ARPA, ADPA, and now STI-R. Is this where streptomycin production finally comes into play?
You got it. STI-R is the final piece of the puzzle. It's the one that turns on the genes responsible for making streptomycin.
Wow, that's a lot of steps. It's amazing how these tiny microbes have such intricate control over their molecular machinery.
It really highlights the complexity of life even at the microscopic level. And remember, this complex dance of molecules isn't happening in isolation. External factors like nutrient availability also influence antibiotic production.
So it's like the microbe is constantly assessing its environment and adjusting its antibiotic production accordingly. It's a survival strategy.
Absolutely. For instance, if phosphate levels are low, Streptomyces might ramp up antibiotic production to compete with other microbes for resources.
It's all interconnected, this intricate web of signals and responses, all fine-tuned over millions of years of evolution. So before we go deeper into the specific mechanisms of how these antibiotics are actually built, I'd like to challenge you with a few questions. Think of this as a mini review session to test your understanding and see how this knowledge can be applied in your work as a medical microbiologist. Ready to put your knowledge to the test?
Hit me with your best shot.
All right, question number 1. What percentage of microbial natural products are estimated to have some form of bioactivity? Think back to that impressive number we discussed earlier.
Yes, that was a big one. If I remember correctly, it's estimated that about 50% of those 70,000 microbial natural products exhibit some sort of biological activity. That's a massive pool of potential therapeutic compounds waiting to be discovered and harnessed.
Exactly. Now for question number 2, we talked about the hidden potential of Streptomyces. What's the estimated potential increase in the number of antibiotics we could derive from Streptomyces alone, considering those cryptic gene clusters?
That's right. If we can unlock the secrets of those cryptic gene clusters, we're looking at a potential 25-fold increase in the number of antibiotics we could get from Streptomyces alone. That's a game changer.
Absolutely. Now let's zoom in on those fascinating signaling molecules. Question number 3. What role do butyrolactones play in the intricate world of Streptomyces?
Well, butyrolactones, or as we like to call them, Streptomyces hormones, are the key players in kicking off the antibiotic production process. They bind to repressor proteins, setting off that fascinating cascade of molecular events that ultimately leads to the activation of antibiotic-producing genes.
Perfectly explained. Now for question 4. We've been talking about these cryptic gene clusters quite a bit. Can you remind us what they are and why they're so important in the quest for new antibiotics?
Cryptic gene clusters are like hidden treasure chests within microbial genomes. They contain the genetic blueprints for potentially novel antibiotics, but they're usually silent under typical lab conditions. Researchers are working on various techniques like co-culturing and synthetic biology to awaken these sleeping giants and unlock their potential. It's a challenging but incredibly exciting area of research.
And it could lead to the discovery of entirely new classes of antibiotics, which is exactly what we need in the fight against drug-resistant bacteria. Now for our final question, let's put all the pieces together. Question 5. Can you briefly describe the multi-level regulatory cascade that controls streptomycin production in Streptomyces griseus?
All right, let's retrace the steps of this molecular relay race. It all starts with A factor, a butyrolactone binding to the repressor protein, ARPA. This binding event releases ARPA from the DNA, which then allows the transcription of the activator protein, ADPA. ADPA, in turn, activates the transcription of STARR. And finally, STARR is the one that flips the switch, turning on the genes responsible for producing streptomycin. It's a beautifully orchestrated process that highlights the power of molecular signaling and gene regulation.
That was a fantastic explanation. You aced all the questions. And I hope our listeners are starting to appreciate the intricate world of antibiotic biosynthesis and how understanding these processes can lead to new strategies for combating antimicrobial resistance. But before we dive into the specifics of how those antibiotics are actually made, let's take a moment to
appreciate just how diverse these antibiotic factories really are. We've got everything from ribosomes, those protein-making machines, to super complex enzyme systems, all working together to build these amazing compounds.
It's like nature has this whole toolkit for crafting antibiotics, each with its own unique set of tools and instructions.
And, understanding these different biosynthetic pathways isn't just an academic exercise. It has direct implications for developing new strategies to combat antibiotic resistance. Take vancomycin, for example.
Vancomycin's a powerful antibiotic, often used as a last resort against those tough infections.
Exactly. And by understanding how bacteria produce vancomycin, researchers are working on engineering new variants that can overcome those pesky resistance mechanisms.
So it's not just about finding new antibiotics, it's about understanding how they're made so we can stay one step ahead of those evolving bacteria. It's like a constant arms race.
Yeah, you hit the nail on the head. And speaking of clever strategies, one of the most fascinating aspects of antibiotic biosynthesis is this concept of combinatorial biosynthesis. Imagine building with Legos.
Legos.
Okay.
Now you've got my attention. Tell me more about this Lego analogy.
Well, think of each Lego brick as a specific enzymatic module, a building block in a biosynthetic pathway. Nature can mix and match these modules, swapping them in and out to create a vast array of different antibiotic structures.
So nature's like a master builder, constantly experimenting with different combinations to see what works best against different pathogens.
Precisely. This combinatorial approach has led to the incredible diversity of antibiotics we see in nature. And it's not just nature that can play this game. Scientists are using this same approach to engineer new antibiotics.
So we can play Legos with these biosynthetic pathways too. That's amazing.
Absolutely. By understanding the rules of engagement, so to speak, we can manipulate these pathways, swapping modules, introducing new building blocks, and essentially creating designer antibiotics.
Designer antibiotics. It's like haute couture for microbes.
I like that. And this field is evolving rapidly.
Yeah.
With advances in synthetic biology and our growing understanding of these biosynthetic pathways, the possibilities are truly exciting.
It's like we're entering a new golden age of antibiotic discovery. But let's circle back to those cryptic gene clusters for a moment. They seem to hold so much promise,
Yeah.
but I imagine there are challenges in unlocking their potential, right?
You're right. It's not a simple task. One of the biggest hurdles is simply identifying these cryptic gene clusters in the first place. Microbial genomes are vast, and finding these clusters is like searching for a needle in a haystack. And even when we find them, figuring out how to turn them on is another challenge.
It's like we found the treasure map, but now we need to find the key to unlock the treasure chest.
Exactly. But that's where techniques like co-culturing and synthetic biology come in. Researchers are making progress in coaxing these genes to express themselves, and the payoff could be huge. Imagine discovering entirely new classes of antibiotics hidden within these cryptic gene clusters.
It's mind-boggling to think about the potential that's out there just waiting to be discovered.
And it's not just terrestrial microbes that hold this potential. Our book also highlights the vast unexplored microbial diversity in marine environments. You see, most of our current antibiotics come from microbes found on land, but the oceans are teeming with life, much of it microbial.
So we've been focused on one small corner of the microbial world, while a whole universe of possibilities exists beneath the waves.
It's like we've been looking in the same backyard for far too long, while a vast ocean of microbial wonders awaits us.
And I bet those marine microbes have evolved some pretty unique ways to produce antibiotics, considering the harsh and competitive environments they live in.
Absolutely. Researchers are already making exciting discoveries in this area. For instance, they've found novel antibiotics produced by marine bacteria that live in symbiosis with sponges.
Sponges. I never would have thought of sponges as a source for new antibiotics. That's incredible.
It just goes to show that we've only scratched the surface of what's out there.
It seems like we're at a turning point in antibiotic research. There's so much more to explore, from the intricate details of these molecular assembly lines to the untapped potential of cryptic gene clusters and marine microbes.
And this isn't just fascinating from a purely scientific perspective. This knowledge is essential for addressing the global health crisis of antimicrobial resistance. By understanding how nature builds these weapons, we can develop new strategies to stay ahead of the evolutionary arms race against drug-resistant pathogens.
So it's about understanding the past, harnessing the present, and shaping the future of antibiotics. And for medical microbiologists like you, listening in this knowledge is absolutely key for making informed decisions about diagnosis, treatment, and infection control.
Precisely. It's about empowering healthcare professionals with the knowledge to fight infection and protect human health.
You know, as we come to the end of our deep dive into biosynthesis, I'm struck by the sheer wonder and complexity of this field. It's the story of incredible diversity, intricate regulation, and almost limitless potential.
It really is. And to capture that sense of awe, we thought we'd end this episode with a short poem reflecting on the beauty and importance of this topic. Ready for a dose of poetic inspiration?
Absolutely, bring it on.
Okay, here we go. In the realm of the unseen, a battle unfolds, where microbes wage war their stories untold. From silent genes to potent cures they yield, antibiotics arise on nature's battlefield.
Wow, that's beautiful. It really captures the essence of this hidden world.
It does, doesn't it? And it reminds us that the fight against infection is a constant battle, one that we need to understand to win.
Exactly. And speaking of understanding, nature holds both the problem and the solution. These microbes are both our adversaries and our allies.
We can learn so much from them.
Absolutely. So as we wrap up this deep dive into the world of antibiotic biosynthesis, I hope you're leaving with a renewed sense of wonder for these tiny but mighty microbes.
And for the incredible potential they hold for medicine.
It's a field that's full of surprises and breakthroughs waiting to happen.
Who knows, maybe one of our listeners will be the one to make the next big discovery in antibiotic research.
That would be amazing. So keep exploring, keep learning, and never stop being curious about the microbial world.
And until next time, stay healthy, stay curious, and keep those microbes in check.